Assalamu alaikum students. In this lesson I will discuss about phases of meiosis. Meiosis was discovered and described for the first time in 1876 by a German biologist Oscar Herdwig. The preparatory steps of meiosis are identical to the interphase of mitosis. Interphase is divided into the same three phases G1, S1, S phase and G2. Interphase is followed by meiosis 1 and meiosis 2. Now I will discuss about meiosis 1. In meiosis 1, the homologous chromosomes in a diploid cell separate and so two haploid daughter cells are produced. It is the step in meiosis that generates genetic variations. Meiosis 1 occurs in two main steps, karyokinesis and cytokinesis. The karyokinesis of meiosis 1 is subdivided into prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is the longest phase in meiosis. During this stage, chromatin condenses into chromosomes. The homologous chromosomes, these are homologous chromosomes, which are apparently same in appearance. These homologous chromosomes line up with each other and form pairs. So this forming of lining up and pairing is synapsis. Each pair of homologous chromosome is called bivalent. Each pair of chromosome has two bivalents and 1, 2, 3, 4 tetrads. The two non-sister chromatids of homologous chromosomes join each other at certain points along their length. These points of attachment are called chiasmata. So here is the attachment between non-sister chromatids and there will be exchanging of segments. In next stage, the non-sister chromatids of homologous chromosomes exchange their segments and this phenomena is known as crossing over. So here you are watching brown beads and blue beads after exchanging of these segments. So it is crossing over and due to this the genetic change occurs in offsprings. After the exchange of segments, the recombination of genetic information takes place. After crossing over, each pair of homologous chromosomes remain as a bivalent. The chromosomes condense further and the nucleoli disappear and the nuclear envelope disintegrates. The centrioles that were duplicated during interphase migrate to the poles and form spindle fibers. The kinetochore spindle fibers attach with the kinetochore of chromosomes, while the non-kinetochore spindles from both sides interact with each other. Two kinetochore spindle fibers attach with a pair of chromosomes. In mitosis, we have seen that two kinetochore spindle fibers attach with one chromosome. So here attachment is between two chromosomes. So here you are watching that after crossing over there is formation of chiasmata and crossing over is exchanging of segments between this chromosome number 1 and chromosome number 2. In metaphase, the pairs of homologous chromosomes align along equatorial plane forming the metaphase plate. So here is formation of metaphase plate. In anaphase, kinetochore spindle fibers shorten. It results in pulling apart the chromosomes of each pair. Since one chromosome is pulled toward one pole, two haploid sets are formed. Each chromosome still contains a pair of sister chromatids. Now you are watching that chromosomes arrive at the pole, and this phase is telophase 1. Each pole now has half the number of chromosomes, but each chromosome still consists of two chromatids. Spindle network disappears and nuclear envelope is formed around each haploid set. Chromosomes uncoil back into chromatin. Cytokinesis is the pinching of the cell membrane in animal cells or the formation of the cell wall in plant cells. It occurs and creates two haploid daughter cells when it is completed. So that was all about steps of meiosis 1. In 1911, the American scientist, that was Thomas Hunt Morgan, observed the phenomena of crossing over in the fruit fly Drosophila melanogaster. After meiosis 1, both haploid daughter cells enter a period of rest known as interkinesis or interphase 2. The interphase 2 is different from the interphase of mitosis and meiosis. There is no S phase, so there is no duplication of chromosomes during this stage. Here is the interphase of meiosis 1, chromatin and a centrosome. Then prophase 1, chromatin condenses, spindle formation, that is here. Prophase 1, there is just metaformation and crossing over. Then there is metaphase 1, formation of metaphase plate. Anaphase 1, in anaphase 1, just meta break and chromosomes pulled. In telophase 1, each daughter cell has haploid number of chromosome. And now the division is in these two cells is shifted towards meiosis 2. Then there is prophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, and there are 4 haploid daughter cells. This meiosis 2 is the second part of meiosis and is similar to mitosis. It is subdivided into prophase 2, metaphase 2, anaphase 2 and telophase 2. Prophase 2 takes much less time compared to prophase 1. In this phase, nucleoli and nuclear envelope disappear and chromatin condenses. Centrioles move to the polar regions and make spindle fibers. In metaphase 2, chromosomes attached with the kinetochore spindle fibers and align themselves at equator. This is followed by anaphase 2, in which the centromeres are cleaved and sister chromatids are pulled apart. These sister chromatids are now called as sister chromosomes, and they are pulled toward opposite poles. In telophase 2, it is marked with the uncoiling of chromosomes into chromatin. Nuclear envelopes reform, cleavage or cell wall formation eventually produces the daughter cells, each with a haploid set of chromosomes. So that was all about meiosis 2. So that was all about meiosis, crossing over and genetic variation. I will discuss about significance of meiosis in my next video lesson. Thank you and have a nice day.